Welcome to the PubMed Daily Review for March 060. Today we scanned 3,150 new open access papers. Here are the top 4 selected for detailed analysis. Up next, our first paper out of 4, a comprehensive functional atlas of ALK kinase domain variants reveals resistance landscape to ALK inhibitors by Hyung-Cheol Oh and Hyeongbum Henry Kim from Pharmacology, Yonsei University College of Medicine, Seoul, Republic of Korea. Let's break down its key contributions. ALK gene fusions are critical drivers in cancers like lung cancer, making them a prime target for tyrosine kinase inhibitors, or TKIs. While these drugs are effective, patients often develop resistance due to mutations within the ALK kinase domain. Many of these mutations are currently of uncertain clinical significance, leaving a gap in our understanding and ability to manage resistance effectively. To systematically map resistance, researchers used a powerful gene editing tool called prime editing in a cancer cell line. They engineered over 3,200 different variants across the ALK kinase domain, covering nearly all possible single nucleotide changes. These engineered cells were then exposed to 3 generations of ALK inhibitors, alectinib, lorlatinib, and zodazakib, to see which variants conferred resistance. The study revealed a detailed landscape of resistance. For alectinib, 45 variants were found to confer resistance, while lorlatinib showed resistance in 75 variants. Interestingly, zodazakib, a newer generation inhibitor, had 56 resistant variants. These findings highlight that resistance mechanisms can be specific to the drug used, with some variants impacting sensitivity across multiple inhibitors. This research provides a crucial functional atlas of ALK variants, significantly improving our ability to interpret variants of uncertain significance seen in patients. The identified resistance patterns offer insights into drug-specific mechanisms, which can guide the development of more effective therapies. Ultimately, this work serves as a valuable resource for clinicians, potentially improving TKI selection and resistance management strategies in ALK-driven cancers. Up next, our second paper out of 4, neural stem cell quiescence is actively maintained by the epigenome by Anna Malkowska and Andrea H. Brand from Department of Cell Biology and Regenerative Medicine Institute, York University Grossman School of Medicine, New York, USA. Let's break down its key contributions. The nervous system relies on a population of special cells called neural stem cells, or NSCs. These cells can enter a state of reversible dormancy, known as quiescence, which is vital for maintaining the nervous system and repairing damage. When the body signals for it, these quiescent NSCs can wake up and start dividing again. But what keeps them in this dormant state, and how is it controlled? To investigate this, researchers used the fruit fly, Drosophila, as a model system. They mapped the epigenomic landscape of neural stem cells, looking at how the DNA's packaging and associated proteins change during quiescence and when the cells reactivate. They specifically examined chromatin accessibility and histone modifications to understand the regulatory mechanisms at play. Surprisingly, the study found that in quiescent NSCs, the chromatin actually becomes more open and accessible. However, genes that should drive cell division were found to be repressed, even though they were in accessible regions. This opening of chromatin was a unique feature of quiescence and reversed upon reactivation, revealing a sophisticated epigenetic control. This research reveals that neural stem cell quiescence is an actively regulated process, driven by significant epigenomic remodeling. This opens up exciting possibilities for therapeutic interventions, potentially allowing us to control stem cell activity for treating neurological disorders or promoting repair. Future studies will be crucial to see if these findings hold true in humans and how we can harness this knowledge. Up next, our third paper out of 4, mRNA-engineered CRISPR-Cas epigenetic editors enable durable and efficient gene silencing in vivo by Chao Su and Jiankang Zhu from Institute of Advanced Biotechnology, Institute of Homeostatic Medicine, and School of Medicine, Southern University of Science and Technology, Shenzhen 518055, China. Let's break down its key contributions. The precise control of gene expression is a major goal in developing new therapies. Epigenetic editors offer a way to do this by changing how genes are read without changing the DNA itself. This could lead to long-lasting effects. However, current CRISPR-based editors are too large to be delivered effectively into the body. This research sought to overcome that challenge by creating smaller, mRNA-based editors. To tackle the delivery problem, the researchers engineered compact epigenetic editors. They used AI to optimize mRNA sequences for better expression and stability. These mRNA editors were then packaged into lipid nanoparticles for delivery. The system was tested in mice by targeting the PCSK9 gene, a key regulator of cholesterol. The goal was to achieve durable gene silencing through targeted DNA methylation. The results were quite impressive. A single intravenous dose of the optimized mRNA editor led to an approximately 83% reduction in circulating PCSK9 levels. This translated to a significant drop in LDL cholesterol, down by over 51%. Importantly, these effects were durable, lasting for at least 180 days. Furthermore, the SF01-based editors demonstrated a better safety profile with fewer off-target methylation events. This study establishes a powerful new platform for developing gene therapies. By engineering compact, mRNA-based epigenetic editors delivered via LNPs, the researchers have overcome significant hurdles in getting these tools into the body. This approach holds great promise for durable and precise gene silencing. While these findings in mice are exciting, further research will be crucial to validate the specificity and long-term safety before clinical translation. Up next, our fourth paper out of 4, single-nucleus RNA sequencing reveals KNAP2+ astrocytes triggering a hypertensive effect in the rostral ventrolateral medulla by disrupting glutamate uptake by Shui Zheng and Dongshu Du from B International Cooperation Laboratory of Molecular Medicine, Academy of Chinese Medical Sciences, Zhejiang Chinese Medical University, Hangzhou, Zhejiang 310053, China. Let's break down its key contributions. Hypertension, or high blood pressure, affects over 1.5 billion people worldwide and significantly increases the risk of serious health complications. The rostral ventrolateral medulla, or RVLM, in the brainstem is a key area for regulating blood pressure. While we know a lot about how neurons in the RVLM control blood pressure, the role of other cells, like astrocytes, in this process, especially during hypertension, is still largely unknown. This study aimed to shed light on this critical gap in our understanding. To investigate the role of RVLM astrocytes in hypertension, the researchers used a combination of advanced techniques. They profiled gene expression in individual cells from the RVLM of both hypertensive and control rats using single-nucleus RNA sequencing. This allowed them to identify distinct astrocyte subtypes. They then used methods like radiotelemetry to measure blood pressure and sympathetic nerve activity, and immunofluorescence to visualize specific proteins. They also performed experiments in cell cultures to explore the underlying mechanisms. The study revealed a significant finding: a specific type of astrocyte, marked by KNAP2 expression, was found to be significantly increased in the RVLM of hypertensive rats. When they experimentally increased KNAP2 in the RVLM astrocytes of normal rats, it led to a substantial rise in blood pressure. Mechanistically, this KNAP2 increase was shown to impair the astrocyte's ability to clear glutamate from the synapse, which in turn made nearby neurons hyperexcitable. This research offers a significant new insight into the complex mechanisms of hypertension. By identifying KNAP2 expressing astrocytes in the RVLM as key contributors, it opens up novel avenues for treatment. Targeting these specific astrocytes or their role in glutamate regulation could represent a promising new therapeutic strategy for managing high blood pressure. This study underscores the critical importance of considering non-neuronal cells when investigating cardiovascular diseases.